I see quite a few MPhil students, given that the dissertation deadline is very soon. I really appreciate the very interest. So it's a pleasure to welcome Mike Sikic, and he's at the Singapore Genome Institute, and before was in Zagreb, where he studied electronic engineering and signal processing, and from 2018 you moved to. So welcome all speakers.
Sure.
Online.
Thank you a lot, Pietro. So yeah, my name is Šikić. I'm originally from Croatia, but now I work at Genome Institute of Singapore. I still keep my position in Croatia when I work in computer science. So I prepared something about our research, and there are two fields we work on. One is related to RNA structure prediction. So I'll tell something at the beginning about this part, and then the second part I will talk more about some ideas related to AI and genomics. So yeah, my background is mostly in algorithms, but 5-6 years ago I decided to replace most of these algorithms with AI. So, but let's just start with RNA structure prediction. So yeah, the first part will be about RNA language model. Our language model, I think, still at the moment is the largest RNA language model. And I will briefly tell you about the idea, how we did it, and yeah, some preliminary results we already published at bioRxiv. So I don't know how good you can see this. So probably all of you are aware of AlphaFold 3. But if you try to use the AlphaFold 3 on RNA, it does not work quite well. So here, AlphaFold 3 is in brown, and it is compared with some other RNA structure prediction models. And as you can see, they are not, this is not much better. And there are some outliers there. So if I remove this one, maybe you can see better. And in some cases, like this R1126 or the next one, AlphaFold is better. But most of these sequences are actually synthetic sequences. So for all other, AlphaFold 3 is not better than others. So RNA structure prediction is a really difficult problem because it is not same as for protein prediction, because for protein you can have tens of 100,000 structures which are resolved. So for RNA, there may be 5,000 which are not redundant. So it is a much harder problem, but from that perspective, it's very interesting. And that we decided to try with RNA, and actually the whole A-Star Singapore really trying to invest in RNA field because the RNA therapeutics is something that we are looking for. So we decided to build some model for structure prediction, and we use many already known models inspired by AlphaFold. But we decided to replace the multiple sequence alignment with RNA language model. The problem with multiple sequence alignment is that it takes a lot of time, and especially for RNA, it is not solved so well. So sometimes if we want to produce the multiple sequence alignment of one RNA, it can last for a week or 2 weeks, just for one. So we had a lot of problems. And then we decided, okay, let us try to build the language model. So our language model, we call it RiNALMo. And the previous language model was RNA-FM, so we used that one as a starting point. And all of these models are actually copies of SMF fold, which was developed by Meta, and it is based on BERT encoder. So we did almost the same. So we use the same masking strategies as BERT and SMF folds, and we tried different masking strategies, but it didn't help. So for RNA language model, we just use each consecutive nucleotide as a token, and we try. So briefly, and after we train the language, we try to fine tune for several downstream tasks like secondary structure prediction, contact map prediction, splice site, mean ribosomal load, or something else. And what was the biggest surprise? We achieved that for the first time, it looked that we can have some kind of generalization. Before, it was some paper 2 years ago, they tried to show that deep learning models cannot generalize well for prediction of RNA secondary structure. In a way that if you train on several families, for example, here are I think 8 or 9 of them, and if you use some kind of leave one out and then try to predict on this family, the results were pretty bad. So the first 2 columns are existing thermodynamics models, thermodynamics-based models for secondary structure predictions. And from here on the right are deep learning-based models. So the RNA-FM is the previous RNA language models, and these 2, MX-fold and U-fold, were designed specifically for structure prediction. So as you can compare results for thermodynamic-based models, they achieved much better results. Also, it's important to mention, and I didn't mention that before, today, for discussed competition, still best models are not AI-based. They're thermodynamic-based with some of human interaction. So it was also thought that probably secondary structure prediction would be the same. But when we screen now and we we just add some resonance and then try to predict this secondary structure, we actually achieve better results. It is interesting that we achieved worse results for one family of RNA, and for this family, the tool that is AI-based actually achieved the best results. But if you do some means, I think it's interesting to see that we outperform other methods, and that there is something that we can learn using language models that can help us in prediction of secondary structure, and in the future, I hope, for tertiary structure as well.
Are you trying to predict non-canonical structures in this? Like RNA-seq has a ton of non-canonicals. Is that due to the description?
We use non-canonical nucleotides or
base pair interactions. So there's like AC pairs all over RNA-seq, or like weird ribosipher things, stuff like that. Are you trying to predict those, or you didn't do it?
So we just tried to do some several available families, and we didn't try some. And at this moment, we don't use
These aren't modified. These are unmodified because that doesn't make all sorts of non-canonical interactions that are important to the structure. So like RNA-seq is modified. And like RNA structure can't do it within parameterized. Are you trying to with your model?
We should not try this model. So we are not experts in RNA, and yeah, we will see if you have some specific tests, you will try. Results are interesting, and now we are working and trying to use some ideas how we can score RNA secondary structure. So if we can find a way how we can score if the structure is good or bad, we can try which was used in our. To understand because we use a very similar model as RNA-FM, but we achieve better results. So the only differences is that they use this learn this position of the coding state as single-fold, and they use the activation function. And we changed use log and Sikic. So we just changed some positional encoding and activation function. But we try to test all of this. So for example, here we have very small RiNALMo model that has 30 million parameters in comparison with RNA-FM with 100 million parameters. We have slightly shorter embedded dimension, and we have the same number of layers in the encoder, and the performance is actually comparable. So we have 3 times smaller model, and and I will show you the several results. So we call this one micro, and we have 3 iterations. The first one is base. It is really like RNA-FM, same positional encoding and same activation function, and then we add ROP and Šikić. So as you can see, for secondary structure prediction, it is this BP DNA RNA data set with known secondary structure or well predicted secondary structure. So we can see that we achieve better results than RNA-FM when we have ROP and Šikić. But even the base model achieved the same results. Adding ROP and Šikić helped, not too much, but helped. Also, we showed again on these same families step by step how it performs in comparison to RNA-FM. So the first one is the base. So just to remind you, the base one is the same as RNA-FM, and but it has less parameters, 52 million compared to 100 million. And so it is slightly worse. But when we add ROP, it helped. And then when we add Šikić, we see that there is a difference. So it depends on the data set. The first data set, if you remember, was BP RNA. And here we did again just leave one family out. And here we needed to add both of these improvement in positional encoding and activation function to achieve the better results. We also tested perplexity, and we can see that increasing the size helps. And it was previously told that for RNA language models, there is no need for bigger models. And as you have seen that RNA-FM, it's about 100 million, but we increased to 650, and we see improvement in our results. So this is you can see how we increase the size of the model. So if you remember, that was the original one when we add ROP and Šikić. Then we increase the size and again, and we achieve better and better results. So it's possible if we have more data And we can even try with larger models. So what we are working now. So the original model was trained on RNA Central. It is a famous database for non-coding RNAs. And now we are adding some coding RNAs from NT, and we found lots of RNAs extracted from microbial data, and there are around 1 billion sequences. So we are training on that. Also, we are trying to add during the learning shape data. So for those who are not familiar, shape data is some kind of experimental way that you can see which nucleotides in RNA are in contact with other nucleotides or not. So it can be, it is the high throughput experiment. So you can produce many of these. And usually this shape data people use when they try to model, especially secondary structure of RNA. So we tried to add them using several strategies, but so far we haven't seen any improvement by adding shape data. Maybe one of the problems was that we use this 2 million RNA shape data created by Rijudas, but most of them are synthetic. So we will try. We have generated at GIS our own shape data, so we will try to see if this can improve the results. Also, the current version takes just 1024 tokens. We will try to increase it to, I think it's feasible, to 8,000, because one of our goals is understanding some potential drug targets, which are non-coding RNAs that can be like MALT1, that can be around 8,000 nucleotides. And yeah, as I mentioned, we are trying to develop some model that can actually predict if the secondary structure is good or not. So some experienced biochemists can really recognize if something is wrong, and we are trying to use their experience to help us to develop some function that can help us to say which structure is good or not in some way, because we would like then to involve this structure in self-distillation. So yeah, that was about our work on RNA structure prediction. Now we have just finished our structure prediction model, but yeah, it's too early to talk about that. But we will involve in this model, RNA language model as input, shape data, but we also have some information, also experimental information that can give us some long distance measures between some helices in RNA. So we will also try to involve that. So just to conclude, RNA structure prediction is much, much more difficult than protein. And in the future, I really believe that for any RNAs longer than a few hundred nucleotides, we will need to try to predict some complexes between RNAs and proteins, and the way how RNA folds, it's still a mystery, but it's a very interesting field. So this is the first part, mostly related to RNA, and I have to mention the team and Rafael is in Zagreb, team is in Singapore, who worked on this. And we have great partners, Roland, who is computational chemist, and Yue is a biologist. She produced all of these experimental high throughput data. And I really believe that the whole team is needed to work on this. So yeah, the next part, it's mostly related to something that I have been working for 10 years. It is the novo genome assembly. So the idea of genome assembly is to try to reconstruct the sequence, but we cannot read. Still, we cannot read the whole sequence at once. So it is fragmented. There's several reasons from that. And because it's fragmented, we need to find a way how we can reconstruct. So you can imagine that you have many books, but the same books. And you can just throw a bomb and destroy it, and you will have so many fragments. But because you have many of them, you can find some overlaps. And then you can try to reconstruct page by page. But this is linear problem, and people have been working on this problem for the last 20 years, or maybe even longer. And an usual approach was just algorithms. And just recently, group of scientists reconstructed the first human genome telomere to telomere. So that means that know each nucleotide in a genome for each chromosome. Before, this was not known. But now we can do this. Okay, there is the one part, it is very interesting, and we are working on that in Singapore, how to reconstruct the last remaining part, which is not exactly known. We know how these segments look like, but we still don't know what is the order and what is the length. They are so-called the rDNA, the part that's called for ribosomal proteins and RNAs, and they are still known. But we can say that most of the genome is reconstructed. Why this is important? This gives us opportunity that we can use AI. For AI, we need a lot of data, and here we need a lot of data. So just briefly, how the genome assembly idea looks like. So we have a genome, we sequence them because there are many fragments. So we can do this fragmentation, but also our DNA is fragmented during the process of its extractions. So we don't know what is the order of these fragments. We need to reconstruct. So the first phase in the novo assembling, we try to overlap. So just like trying if you try to resolve some puzzles. So first we overlap them, and then we use some kind of graphs in a way that each read, each fragment is a node. And if there is an overlap between them, we can represent it by an edge. And in that case, theoretically, if we can find so-called the Hamiltonian path, we can reconstruct the sequence. But this is, again, theoretical, and there are many other problems because of errors in sequencing. There can be some edges that we should not visit, so it is not so easy problem. So the way how do people do this is try to simplify, just to try to remove some parts of graph that are obviously unimportant, and then trying to find solution. The problem with current assemblers is that because of overlaps, missing some parts, it is very, very difficult to reconstruct genomes in the full length. So we have these fragments, we call them contigs, and the final assembly usually have many contigs. Today, it is possible to achieve without manual operation to reconstruction some chromosomes from end to end, but usually sometimes this is not possible because of some problems in overlap, but also in trying to find the right path in a graph. So this is the traditional approach. There are two main approaches how we construct graphs, but this is just a detail. So this is the example that you can see how this graph look like. So this is CHM13. This is this first telomere to telomere reconstruct genome, and this is just one chromosome 20, one of the shortest chromosomes, and it's sequenced by PacBio, one of sequencing technology. So yeah, we need to find a right path in this kind of graph. So what are the problems of the current approach and why I decided several years ago trying with AI? So we developed some of tools for de novo assembling, but from our experience at the beginning, we had just a few genomes that we can use to test. And because of that, we actually manually adapt some parameters to find the best solution. And I'm pretty sure all others do the same. If you look the code, there are many parameters with some strange values, and this is the way how it looks like. But there are so many other genomes, and especially if you have the long plant genomes, which are more difficult to resolve, and maybe one day even aneuploidy genomes that are present in cancers. So we had the idea to not try to use heuristics in a way that we try to tweak until we achieve the good results on several genomes. We just try to leave it to machine learning. So it was obvious because we started with this graph traversal to use the graph neural networks. So what is the idea? Idea is that we have the reconstructed genome, and we know something about the way how people sequence, how sequencers work with some error, so we can simulate. There are already simulators. As we have this gene, we can actually create as many as we want the possible graphs. And in that case, we know what is the exact solution. Because we can always know from which part of the genome each read comes, and we can reconstruct it, and we know which parts are good or not. And we can also evaluate how good we are. So the idea is following. We will try to achieve unfragmented assembly, trying to find the path. So what we will try to do? We will try to predict the probability of each edge, if this edge is good or not. Is there, can we go to this edge or it is forbidden? If we can predict it correctly and we just try to decode, we can resolve the graph. So we are not the first who used this idea. The graph neural networks were used for many different problems, like travel salesmen, and we also tried with trying to find the Hamiltonian path. It's not perfect, but it can achieve some relatively good results. So what we did, we just used a simulator and simulate graphs. And with simulators, we can simulate all kinds of errors in sequencing technology we want. And we generate graphs using some existing tools for construction of these graphs from overlapping reads. And as I mentioned, that we know in advance which paths are good, which paths are not good. And it can be the several good paths. It is okay. And it is interesting that actually number of paths which are not good, it's not so big. It's small, but we need to take care of that. So I suppose at least few of you are familiar with the graph neural networks, and it is different kind of structure. So we actually try to learn using some kind of massive passing algorithm that we pass the information from the neighboring nodes, and that's the way how we can train the model and how we can learn some edge and some node features in a graph. So there are different ways how we can aggregate messages. For example, we have here 5 nodes. The nodes 4 is in the middle, and all other nodes send some messages, and we can use any kind of aggregation which keeps some kind of that it is resilient to any kind of permutation. So we just sum all of them. And at the end, in each iteration, we'll learn the next hidden state of the node, and also at the edge. So the network is quite small and simple. So we have some node features and edge features. There are not so many node features because we don't have so many information about node. So we use some kind of random node features, but for edge features we have. So what we know? We know how the overlap of 2 reads look like. What is the similarity between these 2 reads, because they can have errors? What is the length of this overlap? And we can involve these at some kind of starting features. So we just have a linear transformation so that we have some representation of them, and then we feed that to graph neural network. And then the end, once when we learn the graph neural network, we have some node and edge features will fit to fully connected layers, and we just try to predict for each edge if this edge is good or not, that we can go to this edge and finding the final path. So we use the gated GCN, and there were several other GNN architectures, but we found it suitable, and we slightly adapted that for this purpose. And you can see that many people tried the similar approaches before, but we just slightly changed this. So this is the original idea of gated GNN. So we have these parameters that depends of age representations, and then they define how we can multiply to the matrix. If you want, we can go later to details about all of these matrices. So the major difference, the first one, is that people before just treated graphs as undirected graphs. And here we focus more on treat them as directed, because in assembly graph, we can have some kind of direction. And when decided to treat differently edges that come to the node and edges that go out of this node. So we separate these 2 parts. So we have information before node and information that flows after the node. So this is the one of the differences to traditional way how people represent the graph neural network. And Yeah, we train them and we use classical cross entropy loss and try to predict the final probability of each edge. So we use node features, but in the end, we are just interested in the edge features. And so for each edge, we just add the nodes around this one. Okay, once we can predict each of these edge features, so we can predict the probabilities, and then what we can do with these probabilities. The basic idea is just randomly choose one node and start to traverse to the graph. Then we just go from take in each node the edge with the highest probability. So we just use the greedy approach. And then we stop once when there are no more nodes to traverse. We usually try the several rounds and trying to find the best one. So we use the greedy approach. It is interesting that we tried the beam search and we didn't achieve any better results than with the grid search. Another idea we used is that assembly graphs has one feature that is interesting. So we have two strands of DNA. And if we go in one direction and if we go in another direction in a graph, actually the probability should be the same. So for each of these graphs or each of these nodes, we can imagine that one node is actually two of them, which are virtually connected. So there are two strands of DNA. And we have the next node. It is also two. What we know about the probabilities between them, these probabilities that this is correct edge should be same. And that we just involved, this is the third parameter, that we use this kind of symmetry, that if we try to flow some information in one direction, it should be the same in other direction to add this loss. It is just some kind of the absolute difference between logits of each of these probabilities related for each edge. So we have these two edges and their probability should be same. This opens us a many probability that we can even train the model if we don't know the ground truth, because we can always push them to try to have this symmetry. And this symmetry loss helped us a lot. So what do we achieve? So there are several genomes. First one is a human one. The second one is mouse. That is Arabidopsis, is a plant. And the last one is a chicken. So it is comparison to the best available tool at this moment. It is HiFiSM. I said best because it's the best that uses one of these paradigm is OLC. And we created the graph with the same tool, with this HiFiSM tool. And then we compare their traversal to the graph with ours. So for human, we achieved the better results than HiFiSM. And these results, it's measured in so-called NGA measure. So this measure takes us something about the contiguity of the assembly of the gene. It is interesting that we achieved a much better results on humans, but it's not a surprise for us because we trained humans. We don't train on the same human genome. We trained on another one. but we achieved better results. But what is interesting, at least for me, is that this is some kind of relative values to the maximum possible one. So if it will be 1, all the results will be perfect. So as you can see, the results for human are much better than for others. They are very good for our Arabidopsis. But both of these two are actually known or genomes, and probably people know them very well. And it is interesting for the mouse, results are much worse. So we believe that there is still a lot of space that we can improve and generalize better using machine learning instead of classical algorithms. There are lots of work, but we just started to think. We are probably the first lab who tried this approach. And with new genes that can be reconstructed from telomere to telomere, we can have more data. We will know many kinds of graphs that can happen in future. And we believe this method one day will be better and better, and we can achieve the better results. And I hope some other groups will join us. It's going to be much more interesting, and it will put some pressure. So here are again the same results. Okay, it is not so easy to see them because of my slides. I apologize, but there are many different measures, and results are in many cases very similar to existing tools. And we are quite happy with that because these tools are many tens of thousands of lines of code, and we just use the simple, I say simple neural networks. This is the preprint. At the moment, it's under review, and I hope in the following weeks we'll have some reviews, and yeah, I think it is quite interesting, and we will continue working on this, especially for more complex genomes. And we are aware that there are many problems, and there are limitations that we have this approach, and at this moment we just use on haploid genomes. We don't do any phase diploid assembly, but you need to start. So as CGM13 started with haploid genomes, we also started with the haploid one. So if we can prove this, that it works, we can start working on diploid, and we actually started, and the results are very promising. And what we found interesting that, when you spend some time looking at some problems, at the beginning, it looks very difficult. You don't know. We spent 4 years working on this, and we needed a lot of time to find something that it works. And now we started to analyze more. And it is interesting that there are many nodes and edges that they are actually correct. So there are many ways that you can do things correct, but there are few of them that you can do wrong. And if you go over the wrong edge, you make a big error. So we started to analyze, okay, where are these decision nodes? So if we make correct decision at this node, if we find the right edge at this decision node, it will help us. So we don't need to solve all possible problems. We just need to solve the problems in specific decision nodes. And most of these errors are expected in centromeres. And we found that I think 86% of decision nodes are actually in these kind of regions. We will probably find them in RDNAs, but nobody has resolved RDNAs so far. So we are waiting for this, and we will find how we can solve this problem. The advantage of this method is that once when we have a new data type, when we have new type of reads, we don't need to make major differences in our code and try to find the new parameters. We just simulate again, reads, and use the same strategy. So it is a much faster way to learn in the future. And this work was done mostly by Lovro and Martin, who are PhD students in my group, and with great help from Xavier, Thomas, and Kenji, who help us a lot how to set up this problem. And we are continuing work, and yeah, I hope we will soon have some results. And for the last one, I mentioned to you that maybe in the future we will have some new type of reads. And this is something that we recently finished. And we tried to use some AI to correct sequencing technology to correct reads. So we call it Herro. So 8 years ago, when long read technology was really at the beginning, and especially nanopore, we developed a tool that could correct assemblies. So once when you have this assembly, it's some kind of rough assembly. You can reuse all of reads from the beginning to try to improve. In which way? You just need to try to map all of them to this assembly. And then what we do, we just separate and split them into short windows. And in each of these windows, we do some multiple sequence alignment and try to find the best sequence. Because you use many errors, reads, and at the end, if you combine all of them, you'll see the better results. So it worked before quite well. We even tried this for correction of reads before. But the problem is when you try to resolve genomes that have some diploid genomes or polyploid genomes or high ploidity, you cannot just try to correct everything. You need to take care that you can delete some information that helps you in phasing genomes. So phasing in a way that you can separate the information that come from mother and father. And also what is important in genome assembly is that you have repetitive regions like centromeres. So you have one segment after another, and there are very, very teeny differences between them. It is perfect if you have a read that can span the whole region, but usually you don't. Then you use these teeny differences and try to combine them to overlap. And even if you don't have very, very long reads, you can achieve a sample. So we started with this idea, okay, can we actually error correct while trying to keep this information? So today, if you try to assemble genomes, you need to use the several technologies. You need to have some very, very accurate long reads. And these reads are about 15, 20 to 30 kilobase pairs for one technology. Another one can be 50, 60. And usually people use these reads to construct the graph. There is some other technology, so-called the ultra-long technology. And this ultra-long technology can provide you reads up to 1 million. So if you have these very, very long reads, they have lots of information. But at this moment, their accuracy is 95 or something like that, maybe higher. And current tools use these very, very long reads just to guide them to trying to find a path in a graph. They do not use them in the full potential to build the graph. If you have these very, very long reads and then use them to construct the graph, it will be much easier to solve the problem. So that's the reason why we try to achieve this. And the whole framework is similar except the last part. So the first is when you have some reads, you just need to remove some artifacts due to sequencing. There are some adapters. Then we try to overlap all of them. And then we use this AI method to try to correct. It works for nanopore reads. So nanopore is one of technology, and this day was some kind of London calling nanopore conference, and they have some new chemistries, but the current two major chemistries are R9 and R10, and this tool works for both. It is important to say that this method works for reads which are longer than 10,000 nucleotides, and the quality value is about 10. So we can say the quality value is that the accuracy at around 90%. And the idea is that we try to just to focus on this position that we should either keep because they are informative or try to correct. So the model is not so something special. It uses two convolutional networks to try to extract some features and then feed them to transform. So what we use? We again have these windows of alignment of reads to the read we want to correct. We have their quality values, and then we just find the alignment between this read and all other reads which are involved. And how we can detect potentially informative position. Potentially informative position we detect in some strategy that is used before is the following. So if we have, for example, two potential bases that can be at that position, for example, A and T, and you have some support in other reads. If we have one of them at least in three other reads, we say, okay, this can be informative position. We are taking usually 31 reads. So in 10% of them, if there is a different base, we say, okay, we should be very careful with this position. This is potentially informative position. So in the first neural network, we just, from each read, we extract some information, feed to the second one. In the second one, we are taking the information on each position. And again, we use the convolutional method just to extract this information. But what is important? We only care about the position that could be informative or not. All other positions, we will not try to predict. So once when we have this representation, we just feed them again in encoder transformer and try to predict two things. If the position is informative and if position is not informative, with which sequence we will replace this position? Because it's not informative, we can replace. So we trained the model on just 5 chromosomes from human. And at this moment, we can correct up to 30 nucleotides along indels. So indels are some differences in alignment between sequences. And what is important to say, okay, when we sort out these potential informative positions, what about other positions? We just use majority vote. It is not the perfect way, but this is just the way how to do things. And here are some results. So on x-axis are read quality before correction, on the y are after correction. So you can see that actually we can correct them. So this, for example, on the left is chromosome 9. So this chromosome was not used in training. It is such a good example, but we show the similar results on other genomes, that we improved the quality of reads in a way that we measure this quality when we map to the original reference. So improve is between 50 and 100 times. So we can improve the accuracy. You can see similar results on Arabidopsis genome, and there are some other, so the zebrafish, the CAGM13, and it is important just to mention the CAGM13 is older chemistry, R9 with lower accuracy. And as you can also see, as I mentioned, we use reads which has the quality value above 10. So we still can correct some of them with slightly lower, but results are not good. So here you can see the distribution. So the blues are distribution of read quality before correction, and the orange are after correction. And this is a logarithmic scale, so it is not ordinary scale. So as you can see that the quality for Q20 goes to almost Q40, so it is almost 2 orders of magnitude. So we have different kind of reads. We have diploid reads, diploid genomes. The first one is human. The second one is interesting one. So the second one is the genome of one Singaporean of Indian origin, and we are reconstructing it to telomere to telomere. And the current quality is Q70, and we resolved everything except few chromosomes of these RDNAs that I mentioned at the beginning is difficult, but we hope that maybe we can resolve some more of them.
Can I just ask what you mean by read quality? Is that, so the ones that are above 50, are they more than 100 kb long?
Yeah. Yeah. So, this is the Q value. So Q20 is 99, Q40 is 4 nines or something like that. So, yeah.
But Herro doesn't give you a Q value on its output, so you're just looking at the.
So we map read to the reference, yeah. And that's the way how we calculate. So this is some kind of yeah. Okay. So it is not the output of sequencer; it is this concordance quality value. Here you can see the results for several genomes. So first two are human and diploid. The third one is human haploid, and we have the several other species. And as you can see that using only ultra long reads corrected, and we have extracted the parental information using trio binning, we achieve high values of contiguity, high quality value, and I would say some decent Hamming and switch errors. So this is the detail for those who are in the field. That's the quality of phasing. So ideally, it will be that at the end we have 46 resolved chromosome telomere to telomere, but This is the current results, and these are results for coverage of 40. If we increase the coverage, we see more telomere to telomere reconstruct genomes. And what was really a good results for us is that we resolved chromosome X and Y telomere to telomere almost each time we try to do this. Why is this important? Chromosome Y as a paper, the first human chromosome Y resolved was published a year ago in Nature. Chromosome X was published 2 years ago in Nature. Now, just correction of one type of reads, we can routinely do it. Not with manual curation, just assembly. And just the last thing we found during our work. So I think the data is everything, and without good data, it cannot be done. So that's the reason now we can try to make some assemblers using GNN, because we can have data and we can measure things. So this AG002 is deployed genome, and its quality now, it's I think Q80 or something like that. It's very, very accurate. And what we show here, so when we try to measure error rate, mapping reads from the same sample, from the same cell type, AG0 type, cell types, and try to map them to this reference, we see very, very low error rate. But when we try to map them to other human genomes, we see that actually mean error is around 1%. So mismatch rate is around 0.2, but if you add these indels, the error is around 1%. And we also found the similar differences even if we take 2 haploids of the same organs. So what it is a small sample that we have just a few genomes resolved. So the first one is, I think first one is Ashkenazi, the second one is Caucasian, the third one is Chinese, and the fourth one is Indian. And if we want to train any models, or if we want to do anything, we really need to have original reference from the same cells. If we try to train some models and we have reads from different individuals, we need to be careful because the error rate is around 1%. And then, for example, in our Herro model, if we trained just using some sample and original reference, we cannot achieve the high accuracy because the error is mapping is 1%. So we cannot go back. So that is important to take care when people think, and I believe in the future, maybe in up until the 2030, we can routinely sequence and resolve each of human genomes just assembling, not using that. Yes.
And that confounds it.
Yeah, so the real differences, they are not errors. So this is just if I try to assess the quality, I should be aware that this is differences between 2 human couplets. Yeah, we recently, a few days ago, we published a preprint on bioRxiv, and it was joint work done with nanopore, and Dominic is here, who developed Herro, the Hui has tested it and really true with all this. And as I mentioned, Sergey and Paula from nanopore who helped us, especially Sergey. Sergey was the first author of this first human telomere to telomere reconstruct genome, and his experience was really invaluable for us because we started trying to with chromosome 22. Why chromosome 22 is the shortest one? And then Sergey told us, no, it is acrocentric, it has the RDNAs, it is a mess, no, avoid them. So that's the reason that people really need to know the data. This is funded by AI Singapore together with Oxford Nanopore, and now they just copied the Herro and they rename it, and they will be offered to everyone to try to use it. So thank you all. This is my first visit here, and I try to present many things. So probably some of you were interested in different parts, but yeah, I'm happy to take any questions, and I hope it was at least interesting for you.
Interest of time, I guess we could have one or two questions if people do have it. Are you also happy to stick around for it?
Sure.
For people who want.
My question about the second research presented, you said you have used some graph libraries to represent the data. I was wondering which kind of computation ability you had to manipulate that big graph to use some GPU acceleration.
Okay, so because of the sizes of graphs, we needed to use METIS algorithm to try to do the small components so that we can feed in GPU cards, because the memory is the problem. And we trained on A100 with 40 gigabytes, and yeah, that was the problem. But now, when we are focusing on just important part of graphs, we don't need to use METIS anymore. So it is not necessary to separate. Yeah, but it's a good question.
About the RNA part, you mentioned trying to use shape that didn't improve anything.
Yes.
Can you just say a little bit about how you used it? Was it another input feature, or how did it work in the model?
So, for example, we tried to train the language model in a way, this is the first, in that we replace in each batch. In one batch, try to mask some nucleotides. In the next one, try to mask some the shape values. Because there is the one-to-one, so for each nucleotide you have shape values. So that's the first one. We also tried to add shape data as an additional input to network, but it also didn't help. So we're still thinking what's going on, but with or without shape data, they do not make results worse, at least not much worse, but they do not help. So yeah, to be honest, I'm not sure why. We are trying to understand. So the first thing is we found, okay, many of shape data we have from this Ribonanza competition from Regions are actually from synthetic RNA. So maybe that could be the problem, because the rest of sequences we have from RNA Central are actually real non-coding sequences, yes.
In your analysis of RNA structure, do you look at conformational changes? RNA is very different.
Yeah, we are aware of this, but we just use the training data sets from other people, and the flexibility of RNA, it's a huge problem. We are aware, but you need to start with something and try and keep some known. But yeah, we are aware of this.
Perhaps to conclude, or did anyone have any very confident.
That I'm using for Herro, the first step. How do you get that? What's the backbone and how the other sequences? Is that just missing or healing you?
So we just align them using Minimap2, and this is actually the bottleneck.
That's all versus all.
Yes.
But we get pairs of them, and we have to have a file up to figure out some experts and do not agree. So what I don't understand is how from pair to pair we get to the phylum step. So we have to have some backbone. Is that the reference or is that the longest read?
Because we try to correct each read. So that read is actually the backbone. Yeah.
But can it be sped up by when you try to correct one read, you had some information of what made sense and what not. And another read that overlaps this one can make use of this information.
So we use that information that we keep this alignment, so we don't need to align twice. And maybe in the future, maybe we can add the several rounds of correction. Yeah, it's a good question. And we found that sometimes we still can overcorrect some positions. And it is interesting that these positions are usually, of course, homopolymers. So yeah, but I think that there is a space for improvement. But at this moment, if I will improve Herro, I will do better and faster auto alignment than Minimap2. And Dominic and I had discussion during the morning. So we developed the graph map as the first tool for mapping reads for nanopore, and the error rate was huge, so it was 25% or something like that. And then it was developed, Henk Lee developed the Minimap much better than us, as always. Henk is the god of bioinformatics. But still now we have Q20, almost Q20 reads. And we also tested this Herro on Q28 simplex reads, and we still see improvement. And so if we have Q28 reads, do we really need all of this chaining and something like that? So I think we can think about the better, the algorithms that are more tailored for more correct reads.
I think you could explore GNOME aligner, because that's an order in the algorithm, so that'll be faster.
Yeah.
He also designed it for all versus all assembly.
Sure.
So, yeah.
Which line is that?
The aligner.
Yeah.
The aligner.
The aligner.
Liner.
Yeah.
It's GNOME. It's a public repo.
But that's for short reads, right?
No.
That's actually used for this function. That's important.
Yeah, he developed it a long time ago.
Yes, I remember.
So it was designed for long reads.
And one of
It's got a slightly funny database.
we even try using AI just to do all versus all, just compressed reads in some representation, and it actually works. And then if you have a very short representation, all versus all, it is not so difficult because you can use this quadratic complexity because it's not too big in that case. Yeah, it's too many things, and now we're working in two fields, so yeah. Yeah, if you have any questions, please, we can chat. Tonight we are flying back to Singapore. I hope you'll be the better luck. And yeah, if you have questions, yeah. Okay. And I have to thank Chaitanya for inviting us.
And thank you for joining online as well.